In this lecture, we'll be going over the concept of the p-value. We'll be reconsidering how we use this tool. This lecture is premised off of the article Moving Toward a World Beyond p less than 0.05 from the American statistician, authors Wasserstein, Sherm, and Lazar. If you have not read this, please pause, go read it, and come back. The two primary topics of the lecture today are the structure of null hypothesis test statistics and the atom approach to how to use p-values effectively. In order to understand how to use p-values effectively, you need to take a step back and really understand what a null hypothesis test statistic is. This has been a topic of conversation in psychology and neuroscience for a long time. In particular, there's been a focus on dealing with what's known as the replication crisis. The replication crisis is a problem that emerged in the field where uncontrolled sources of variability can lead to dramatically different findings, even when using identical methods. For example, in this article by the Open Science Collaboration, authors tried to replicate a set of well-known psychological findings from a variety of areas in psychology, including social, developmental, cognitive. And they found that they could only replicate a small subset of findings from prior studies using the exact methods. There have been other attempts beyond this paper that have tried to use the exact tools. So for example, use the exact same experimental scripts, exact same analysis routines. And really what ends up happening is that only a subset of findings are found to be replicatable in terms of finding statistically significant effects. In general, there are strong trends towards when things aren't statistically significant, the effect sizes go in the same direction, but by and large, it's been difficult to consistently replicate known findings. Now, this problem rests almost solely on the use of null hypothesis test statistics in our field. In order to understand what a null hypothesis test statistic is, you have to know what the null hypothesis is. This is something that we have discussed in prior lectures, but we're going to go into detail here on it. Remember that the null hypothesis is the probability that you would observe your data if your predicted relationships are not true. This is sometimes chance, but not always. So if you think two variables are related, the null hypothesis is that they are not. Now there's a world where your null hypothesis is true. So you will create many variants of your experiments, and the null hypothesis is true, and there will be a range of effects that you will see if the null hypothesis is true in that world. So you might run an individual experiment and get an observed effect that sits somewhere on this distribution, so that everything to the left is the probability of, everything to the right is the probability of getting the data that you have, call it y, given that the null hypothesis is true. And the p-value is the probability threshold that you're willing to accept of this false positive case, where you are rejecting the null hypothesis. You decide not to accept the null hypothesis, even though it is true. The farther out on this distribution you sit, the less likely it is that the data that you observed comes from a null hypothesis, or a world where the null hypothesis is true. This is the fundamental structure of null hypothesis test statistics, which we often shorten as NHTSs. Null hypothesis test statistics return what we call the p-value. The p-value is just what I showed you on the last slide. It's the probability that the data you observe comes from a world where the null hypothesis is true. So it's the probability that you'd get the data that you have given that the null hypothesis is true. We typically evaluate this by comparing a set of test statistics. One where you have a test statistic where the null hypothesis is true. So you have a world where the null hypothesis is true, and you compare it to the test statistic you get with your observed variables. And if the probability that your test statistic comes from the null distribution, if that's a high probability, then you don't reject the null. If it's a very low probability, then you do. So in a sense, what you're saying is that there's this null, and it produces, you can sample data from this null distribution, and it produces a given test statistic. And if you did that many, many, many times, you would have a distribution of these test statistics that are generated from a world where the null is true. And what you're comparing is the test statistic that you get with your actual data against this hypothetical distribution in a world where the null is true. That's how a null hypothesis test statistic works. That's what your p-value is. It's telling you how likely is it that you get this particular statistic with this set of observed data, given that the null is true. Now, there are many problems with null hypothesis test statistics and the p-value in general. Some of these limitations include the fact that it's dependent upon unobserved data. So p-values are often calculated based off of hypothetical test statistic distributions. And this is actually true even when you use permutation tests, which you've discussed in prior lectures. Null hypothesis test statistics also depend on subjective intentions. They have a very rigid structure that assumes a particular intent. So this leads to things like p-hacking, where you will keep getting new data until you see a p-value that crosses your threshold. An individual p-value or test statistic itself is just a tool, and it will give you the results that you put into it, but how you use it matters. In addition, p-values do not quantify statistical evidence. They're existential tests. They are a limited test that basically says, does the null exist or not in this very particular case? And existence is not the same thing as evidence. This is why you can't really compare two statistical tests that return two different p-values. If you have a p-value of 0.04 and a p-value of 0.01, while those are different numbers and they can be evaluated against different thresholds, the difference between those two p-values is not really meaningful in terms of evidence for or against the null. Really just where they sit with regards to a priori threshold or a prior threshold is what's important. And finally, p-values are just generally poorly understood. So for example, having a p-value of 0.051 is not really that different from having a p-value of 0.049. But because one sits on the other side of our p less than 0.05 threshold, we will say that a result that returns the value of 0.049 is significant. But it's actually not that much different than one that sits on the other side, 0.051. People don't often really capture this. And, we will say that results that have a p-value of 0.051 are, quote, trending, unquote, to being significant. But technically that means that results where you have p0 0.049, which is on the other side of your p threshold, are trending towards insignificance as well. As a result of this relatively poor understanding of what the p-value is reflecting, you see many cases where conclusions are drawn on research data that are actually not reflective of what the p-value is saying. These limitations have led to many poor practices with null hypothesis test statistics in the field. It's important to point out or to remember that null hypothesis test statistics, otherwise known as p-values, are simply meant to be a flag to indicate whether results warrant further inspections. They are a beginning, not an end. whether a result is statistically significant or not really just tells you whether or not you want to dig further into understanding the effect. But that's not how they're typically used in the field, and that is what that has what's resulted in a lot of the problems that we see in the wild. So for example, common poor practices with p-values include concluding solely on the statistical significance of a particular result, whether or not it sits to the left or the right of 0.05, and making that your sole decision. And this is a poor practice when it comes to using null hypothesis test statistics. Believing that an effect exists simply because it is statistically significant. If you have a p-value of less than 0.05 and you then put blind faith that the effect really exists just because of that one single value, you might miss complexity that could lead to a false positive. Conversely, if you believe that an effect does not exist because p is greater than your alpha, let's say 0.05, then you might actually reject a true effect. It might be weak, you might have an underpowered data set, you might have data that has a distributional quality or outliers that might impact your test statistic. But if you just look at the p-value itself and see that it doesn't cross your threshold and decide that the effect does not exist, you'll miss these ambiguities and meaningful details in your data. Finally, a common misconception that has led to a lot of problems in the field is the belief that the p-value gives you the probability of chance alone. If you see a p equal 0.1, people will sometimes say that means there's a 10% probability that the results that I had were generated by chance. And that's not what the p-value is saying at all. And that's an improper inference as to what the p-value is actually reporting. Now, based on these limitations, Wasserstein and colleagues in the article that was the premise for today's lecture proposed taking what's known as the atom approach to using p-values. There is a common trend for many people to criticize the p-value, want to get rid of the p-value, but Wasserstein and colleagues make a valid point that the p-value is a very useful tool. It's not something that should be rejected outright in the field. It's just something that you should use in the limited manner with which it's meant to be used. And adopting the atom approach is a way of using the p-value as part of a whole set of tools in your data to make proper inferences on your data. So ATOM is an acronym that stands for accept uncertainty, be thoughtful, openness, and be modest. Wasserstein and colleagues argued that these are your first principles for using null hypothesis test statistics. Let's go into each concept individually. The concept of accepting uncertainty means that you should acknowledge that uncertainty exists and test for it. That means you want to seek better or more comprehensive measures of uncertainty in your data. You want to basically always measure uncertainty. So along with taking a p-value, estimate your confidence intervals on your effect. You want to decrease focus on data quality and experimental design. This is the adage, poop in, poop out for a particular statistics. If you have poor quality data that goes into a test statistic that returns a p-value, well, that means that you're going to have a poor utility for that p-value. So think carefully about the nature and the quality of the data itself, the experimental design that went into generating the data, when interpreting whether or not an individual p-value is greater than or less than 0.05. Also, by accepting uncertainty, that means you want to move away from and just get rid of the false certainty that you get with calling something statistically significant. Move away from the idea that the p-value is an oracle of truth. The p-value gives you a sense of whether or not a null hypothesis could be true, or whether the null hypothesis exists or not, but it is not the end-all be-all measure of that truth. So even though you can talk about something being statistically significant, that shouldn't be shorthand for real or truth. along with accepting uncertainty, you want to be thoughtful about your data. You want to develop a holistic approach to inferring your data, not just relying on that single test statistic. Look at the whole story. You want to be mindful of the data quality at all stages. This is why earlier lectures focused on tidy data organization and data cleansing steps. You want to have a sense very carefully of what your data quality is. And this goes back to that accepting uncertainty of poop in, poop out, right? You want to know and be very careful of what's the best data that you can put into your test statistic, and really how reliable does that make your interpretation of the effect. You want to consider context and all prior evidence. Every experiment doesn't exist in isolation, or at least a vast majority of experiments don't exist in isolation. There's lots of prior knowledge that you can use to govern your expectations, either the direction of an effect, or its effect size, or its existence. So your individual test statistic doesn't live in isolation, and it should be interpreted in the broader context of all the other evidence that's out there, both across other studies and within your own study. You also want to look ahead to how results predict future outcomes. So you have an individual test statistic, you have an individual p-value, you have an individual regression coefficient, you have the confidence intervals. What does that necessarily mean? If I have a p-value that doesn't cross my statistical threshold, that prior work says should be there, and I have an effect size, let's say a regression coefficient, whose magnitude and direction are consistent, but whose confidence intervals are very big, well, this tells me that I need to think about collecting possibly more data, that maybe I have an underpowered study, and that's why I am getting a non-significant p-value. You want to be very mindful of your effect sizes as well as the statistical significance. The effect sizes are really the more meaningful measure in many cases. So examples of this are like behavioral genetics. In behavioral genetics, the reason those studies require thousands or tens of thousands or hundreds of thousands of samples is because the individual effect of an individual gene on behavior is very, 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 very small. So even though you might get a p-value of less than 0.05, when thinking about what it means for an individual gene to be linked to an individual behavior, the relative magnitude of that effect is going to be functionally quite small in most cases. And so even if you have a significant p-value on an individual effect, you want to interpret that effect in the context of the effect size itself. You want to consider both mechanisms and possible confounds. When interpreting everything together, remember, false positives sometimes occur. Also, false negatives sometimes occur. What are reasons that could contextualize your individual p-value or your individual test statistic and lead to it not being an accurate reflection of the actual relationships of interest? You want to think of your entire toolbox of methods. The p-value is just one tool. Your test statistic, a regression coefficient, is another. The confidence intervals are another. We'll go over methods like Bayes factors in another lecture. All of these things all work together to look at a single effect in different ways. And you want to think about the p-value as just being one tool in that arsenal. And finally, you want to effectively communicate your uncertainty in different ways. And this comes into the writing part or interpretive part of working with null hypothesis test statistics. You want to effectively take all this information, this holistic information, the context, the values of the other metrics, the data quality. You want to take all of that into effect when you're communicating and interpreting the inferences from your results. If you don't, you might know them, but the audience doesn't, and they will very likely make false conclusions or interpretations of your data. Also, Wasserstein and colleagues argue that you should adopt a value of openness. Basically, adopt open science practices, which have been a focus of previous lectures. Release your code. Release your data. Communicate your methods clearly and completely. This is why we focus on using things like Jupyter notebooks or Markdown notebooks, because you can communicate everything together with your code and your data and your inferences all in one clear, complete, efficient way. You want to focus on maximizing the replicability of your result. As much as possible, do everything you can to allow for replicating your results, which means reporting significant and insignificant results. P-values that are passing your thresholds and those that aren't. That's important because this selective reporting of your results leads to what's known as the file drawer problem. A file drawer problem is where only statistically significant results are published and insignificant results or non-significant results are not published. So when an effect is not real, what ends up happening is that if you look at the relationship, say, between the confidence of an effect, which is an estimate of its variability, and effect size, let's say you plot individual studies or individual analyses based on what's reported in the literature, what you'll see is they'll sit on often this other side of this p less than 0.05 threshold. There will be very few studies that will report a particular effect size for a result inside this cone. Now, a real effect, because of regression to the mean, will sometimes be statistically significant and sometimes won't. The more times you kind of point out that studies sit outside of this cone of significance, the more likely you're going to be seeing basically selection bias. As you increase variance and you increase, if you increase the variability of your results, which means going down the y-axis here, along with increasing the effect size, what you'll just generally see is that result often just sits just outside of the p less than 0.05 threshold. Whereas real effects will vary in their size and their significance based off of a host of factors. But if you're only publishing, or mostly just publishing, statistically significant effects, and you look at the literature, you'll miss those non-significant effects. They'll all just sit on the outside of this cone. And that becomes a problem because the file drawer problem means that people have false confidence in a result because they don't see the number of times that it's not significant. Good example of this are effects like the Mozart effect in developmental psychology. It was an old effect from the 90s that said that if you play classical music, in particular Mozart, to babies, they will do better in cognitive tests later in development. One study showed that, and there was nothing wrong necessarily with the study. It was just a false positive. 20 other studies couldn't confirm it. 20 other studies found null hypotheses, and they all had to bundle together into a big study to show that this didn't exist. But it was a big push to show it because those null hypotheses, those null studies with null effects were not being published. Everybody just saw the one study where it was published. So that file drawer problem led to a lot of research in an area that actually had no real effect. Now, false positives happen, so that's not to say that there's anything wrong with the study that publishes a false positive, but we should interpret our reliance or belief in a particular effect based off of its consistency in replication. And the file drawer problem causes a bias in that. Final step of the ADAM approach is to be modest. Being overzealous and inflating conclusions is only effective in the short term. There's no such thing as a perfect study. We believe effects in the aggregate is when they're replicated, when other labs in other parts of the world can replicate it. So there's not a single study or a single paper that will answer an entire question, no matter how small. Inflation of conclusions reduces replicability. So if you say that this is a perfect study and you inflate the significance and size or magnitude of your results verbally, it reduces the replicability because people go into trying to replicate the work or work with those findings or work with those effects, expecting the effect size to be much bigger. You want to be a neutral judge of your findings. You want to temper your conclusions. You want to say why a statistically significant effect might actually be a false positive. You want to say why a statistically non-significant effect might be a false negative. You want to encourage as much as possible others to replicate your work, and this is the open science practices. You want to give your data. You want to have others work with your data, work with your methods, and show that they could do the same thing. Being overzealous and inflating your conclusions really is just a short-term strategy. So when you are a neutral judge of your findings, when you temper your conclusions and you encourage others to work with your results and you show where they might be imperfect, you are playing a long-term strategy. You're trying to give others and yourself, future self, places to look to really test this effect meaningfully. And without it, you might get a really good high-impact paper. But in the long term, if people can't replicate it, you've not only have an effect that just disappears over time, and now, 20 years in the future, nobody believes it, but you also can waste other people's time. You can have people believing strongly and putting in significant time and energy and resources into something that's not real because of this aggrandizement. So aggrandizement means, or is a means of impairing the confidence in your findings. So adopt the atom approach when using p-values. P-values themselves are just a single tool in an arsenal of many tools that look at your data in different ways. It's just giving you an estimate of the existence of a null hypothesis. It gives you one way of gaining deeper insights into your data, but it is not an oracle. It is not a test of truth. It is simply a tool, and one of many, that provide a holistic understanding of your data set. And I hope that's the take-home message that you get from this lecture and the Wasserstein reading. The p-value is a very useful existential test of your null hypothesis, but it should be used in conjunction with many other statistical tests to draw reliable conclusions from your data.